Hi Hires, Metabolism and Survival Unit. We're looking at growth phases, in particular in microbe culture. Okay, so you're expected to recognise this growth curve. Okay, growth curve is made up of 3 stages in terms of growth and then 1 in the decline. So we have the lag, we have the exponential, we have the stationary, and then we have the death. And this is also the exponential phase can also be called the log phase. So lag, log, stationary, death. Okay. This is pretty much it. We're going to go through each of these sections. Okay, so lag phase, this first bit here. Basically what's happening here is we've got a slow increase in numbers. So for a start that looks very small, like 1 doubling is only 2 and 2 is doubling of 4. I mean, once you're up at the 10,000 doubling 20,000, that's a big jump. But what's happening here is we are getting all of the enzymes that are needed are induced. So all of the transcription and translation of enzymes that are required for all of the conditions that it finds itself in. So this is your lag phase where they're getting ready to go. The exponential or the log phase, that's when they're going. They're going at full pelt. So you've got increase, rapid increase. We've got maximum growth rate. So if this is a bacteria that doubles at its maximum rate at 20 minutes, then it's all at 20 minutes. And they're doubling every single time. Nothing is limited, no competition, just zipping up through the numbers. Phase 3, stationary phase. So at this point we have a balance. So for every cell that is being produced, a cell is dying, and we have reached a point in the population where it's just sitting and at a fairly high level now. Now at that point you're getting something that is really important to us. We start getting secondary metabolites. Now secondary metabolites are things like antibiotics. That's where they form. So we need to get the culture up into stationary phase, but not really wanting them to go into the death phase. So it's a tricky balance. We want them past the kind of go, go, go stage of the exponential, but we want them to sit at the stationary as long as we can. Other things are produced in the exponential we want, so it's a balancing act. We then hit the 4th stage, the death phase. So in that one, we start to have a problem with the culture. So that might be that you've got toxic waste products have started to build up. It might be that we're just running out of stuff. They've not got enough food or basic substrates. But what's happening is that the death rate is now overtaking the cell production rate, and so the population starts to fall. So we need to be able to deal with graphing this, and the problem is that the scale does not work nicely. So if we do our kind of normal thinking of, here's your scale. So if we had a 20 minute generation time, and then you're only 4 hours into the culture, and it's getting really hard to plot these numbers on a linear scale because you had to have 1, 2, 4, and you also had to have 1,000 and 2,000 in there. None of this works nicely. So the solution is to use a log scale. And what you're doing there is each division is a power. So you see this an awful lot on microbial growth. Okay, we have what's called a semi-log scale. So what that means is that on the x-axis you will have time, say, and if it's going in minutes it will go up in just 10, 20, 30, 40, 50, 60, a normal linear scale. But the y-axis going up will be a log scale. So here is semi-log paper. Okay, so what you can see here is here's 1 to 10. So we consider that's the first division. But now we have, here's 10 to 100, and there's 100 to 1,000. So each time you've gone up, you've started to go up in another level of a power. Going along the way is nice, simple, straight, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, normal scale up this way. This one is actually showing correct lines. Okay, so you'll notice that the gaps even get smaller as you go up. But sometimes you're only given a main points on the scale, so you might just be given 1, 10, 100, 1,000. So you have to look out for them and be sure that you know what you're looking at there. Okay, so what we have here is a log scale, but you'll notice it's not giving it in a nice way. It's not saying 0, 1, 100, 1,000, but really that's what that is. Okay, here's 1, that's actually 10, and that's 100, because this is just 1 times 10 to the 1. This is times 10 to the 2, 10 to the 3, 10 to the 4. So what you've got here is an increase by power. So just really be careful when you're looking at them, that's all. So you can have, this would be a linear graph, both axes have linear scales. This would be a semi-log. Okay, this is also semi-log, but you're unlikely to see this one. This is going in the other direction. And this would be, just to be pretty, this is a log-log scale. You are not going to see that. So what you actually graph in this is going to be how many cells you really have. We can do this by a direct cell count using things called hemocytometers. We could use colorimeters just to measure the density of the cells just by absorbance. We can also look at viable cell count. So instead of just looking at how many cells are there, how many of them are actually living? And that might be a different one that comes up the side. And for that one, quite often what we use are dyes. So we look at dyes that are only taken in by specifically some living cells, or dyes that are only taken up if you no longer have a functioning cell membrane. Okay, that's that.